welcome back to the continued part of microbes in human welfare. Now, in the previous class, we had came to the part of microbes in household food products. Second one is microbes in industries. Now, let's continue the part of microbes in industrial products. In the previous session, we came across the first product where we call it as alcoholic beverages or fermented beverages. The second part is regarding the antibiotics. The third part is regarding the chemicals, enzymes and other bioactive molecules. So let's study regarding the part of the antibiotics here. Now regarding the part of the antibiotics, already we know we are using it for ourselves and we use it as a medicine. Okay, now what exactly the antibiotic means? Now antibiotics, now this has been taken from the Greek term. Anti refers to against and bios means life. Look at the heading. Antibiotics. Anti refers to against, bios meaning life. That means these are the substances that are produced against the life. Against the life means whose life? Our life? No. It is against the life of pathogens, against the life of microorganisms which harm to the humans. So that's why the name antibiotics. So antibiotics are the chemical substances that are used to kill the microorganisms which harm the humans. So that's why the name antibiotics. Okay. Now regarding the part of the antibiotics, how can we define antibiotics? Antibiotics are the chemical substances that are used to kill the microorganisms. So that's why antibiotics can be defined as these are the chemical substances which are used to kill microorganisms, or let us call them as pathogens. Okay, so this is regarding the definition of antibiotics. Okay, then apart from this, the question comes, who discovered the first antibiotic? The question comes. As you are familiar with the person who discovered it, his name is Alexander Fleming. Okay, the question. The question what comes here is, did Alexander Fleming actually discover antibiotic? No, he did not discover the first antibiotic. He accidentally discovered the first antibiotic. Okay, then how did he accidentally discover his experiment? Okay, the antibiotics in his experiment. Now, Alexander Fleming, let us take his name. Discovered the first antibiotic as a penicillin. As we know, penicillin was the first antibiotic that was produced by Alexander Fleming. So he discovered the first antibiotic and named it as penicillin. Okay. Now, how he accidentally discovered his penicillin? Now, Alexander Fleming, he was working on an experiment. In his experiment, he used certain microorganisms, especially certain bacteria. And the bacteria which he has taken in his culture medium, this is culture medium, consisting of a bacteria. The name of the bacteria is a Staphylococci. Similarly, Staphylococcus. Look at the name. He was doing experiment on the bacteria called as a Staphylococcus or Staphylococci bacteria. Now, in his culture media, on the side, there was development of certain modes. On the sides, there was development of certain modes. Mode refers to fungus. So some fungus was growing here. Now, when this mold was growing, this mold did not allow the growth of Staphylococci bacteria. That means this mold was killing Staphylococcus. He called this mold as an antibios. Bio means this is the life. Anti means this is the fungus that is growing against the life of his experimental organism, a bacteria called as a Staphylococci. Look at the discovery. Look at the accidental discovery. So this did not allow the growth of a Staphylococci, and hence when this was identified as a mold, and later on he came to know that this mold is nothing but it is a fungus. Then what was that fungus? This mold, that fungus was Penicillium notatum. This was the fungus. Look at this. Penicillium notatum. So Penicillium notatum was the fungus, was the mold which was growing in his culture media. It did not allow the growth of Staphylococci, and hence he named it as antibios. Later on, the name was given as antibiotic. Okay. The name of the fungus is called as Penicillium notatum, and from this the antibiotic penicillin was discovered. Now it is widely used to treat many of the diseases. Okay. So in this way he discovered it. Now, after the discovery, he did not directly name it, but its therapeutical value, its antibiotic value was explored by two more persons. One was Howard Florey and Ernst Chain. So two more persons were there who were responsible for the showing the therapeutical value of the antibiotic. And let's look at their names. The therapeutical properties of antibiotic was shown by Ernst Chain and Howard Florey. Okay, the therapeutical property, meaning the antibiotic property, was shown by these two persons. One is Ernst Chain, the second one is Howard Florey. Now, when they showed its antibiotic property, that antibiotic was widely used for the soldiers who were wounded in World War II in America. So this was used. The antibiotic was used to treat the wounded soldiers in World War II. Now, when the first antibiotic was identified, when the first antibiotic was observed, and when its therapeutical value or treatment value was identified, then this antibiotic was given for those soldiers who were wounded in World War II, especially the American soldiers. Because of the saving of life of many soldiers, these three persons have been awarded with the Nobel Prize in the year 1945. Okay. So here what we come across, fluorine, chain and flame were awarded with Nobel Prize in 1945. So fluorine, chain and flaming. These three persons were awarded with the Nobel Prize in the year 1945 for not only the discovery of the antibiotic but for saving the life of many soldiers who were wounded in World War II. So that's why this grabs the attention of many of the scientists who later on discovered the antibiotic. Now if we look at the names of different types of antibiotics, almost if we go through its list also, we find many different types of antibiotics, and those antibiotics are tetracycline, erythromycin, kanamycin, azithromycin, so azibest. So in this way there are around more than 1,000 different varieties of antibiotics that are discovered. Okay, and hence it grabs an attention. And remember, the first antibiotic which was discovered was penicillin. It was discovered from a fungus called as Penicillium nodata. So penicillin was the first antibiotic. Now from many bacteria, lot of antibiotics have been produced, and these antibiotics are also effective against various types of diseases also. Okay, so what are those diseases? Some deadly diseases like leprosy is there, whooping cough is there, kala azar is there. So those diseases through which it could have killed many persons, because of the discovery of these antibiotics, it is saving the life of humans. So that's why antibiotics grabs its attention. Okay. Now regarding the part of the antibiotic, so this is as far as your NCERT part is concerned. Now if we want any additional name of antibiotics, we can go through it also, and the list I am giving you an additional list of the antibiotics along with the microorganisms from which they are produced, because antibiotics are produced from microorganisms only. So how they are produced and how they are used. Okay, so regarding that part, so I will give you an list. Okay, it is an additional part out of the NCERT. So let's look at that part. Okay, now here we have the list of all the antibiotics from which they are produced. Okay, now let's look at them one by one. Look at the first antibiotic. The first antibiotic discovered was penicillin, from Penicillium notatum. Look at the rest of the antibiotic. Erythromycin, it is the antibiotic that is produced from Streptomyces erythroceles. Look at the part. Erythromycin is produced from a bacterial series, Streptomyces. Aureomycin, it is the antibiotic produced from Streptomyces aureofaciens. Okay, this is the microorganism from which this antibiotic. All these are the list of antibiotics. Terramycin produced from Streptomyces rhamnosus. Chloromycetin antibiotic produced from Streptomyces venzile. Streptomycin antibiotic produced from this microbe, Streptomyces griseus. Actinomycetin produced from monospora species. Tetramycin, Streptomyces griseus. Neomycin, which is antibiotic produced from Streptomyces fridii. Kanamycin, an antibiotic which is widely used in treatment of disease also, is produced from Streptomyces and Kanamyceticans. bacitracin antibiotic produced from Bacillus licheni forms. So look at the list of these. So here I have given you an additional list of the antibiotics and the microbes from which they are produced. Now here what we have is majority of the microorganisms are bacteria from which the antibiotics are produced. But the first antibiotic was produced from a fungus. Okay, so this is regarding the additional content of the antibiotics. So as I told you, today more than 1000 different types of antibiotics are discovered. Still we need to discover an antibiotic against the present day virus, the coronavirus. Okay, so let's hope for the best. Let's discover the antibiotic as soon as possible and let us eradicate the disease from the world. Okay, so with this we'll move on to the next part. This completes regarding the part of the antibiotics, and we are moving on to the third title, what is chemicals, enzymes and other bioactive molecules. Okay, now the third heading is divided into three parts. We just divide it into three parts. The first part is chemical, second part is the enzymes, and the third part is bioactive molecules. The third title says chemicals, enzymes and other bioactive molecules. Okay, let us split into three parts, 1, 2 and 3. The first one, chemicals. Let's look at the different types of chemicals, then enzymes and finally bioactive molecules. So let's come across the heading of chemicals. As we know, chemicals are produced in industries. In what quantity they are produced, whether they are produced in small quantity or whether they are produced in large quantity. Yes, they are produced in large quantity. As we know, in the previous session we came across the part of fermented beverages where the alcohol is produced in larger tanks called as fermenters in large quantity. Almost lakhs of liters of alcohol is produced. Similarly, the chemicals are produced in large quantity. What is the necessity of production of these chemicals? Because the chemicals are used in all the laboratories, not only in the PUC laboratories, in degree laboratories, in the PGs also. It is used in the research labs. It is used in the field of medicine also. Okay, in the all the lab technicians require the medicine, the chemicals, and these chemicals has to be produced in large quantities and it has to be supplied throughout the world. Not only in India, it has to be supplied throughout the globe. So that's why the chemical has to be produced in large quantity and hence it forms the part of an industrial product. Okay. Now let's study few chemicals. So there are 4 chemicals which are given for you. Let's call them as ABCL chemicals. Okay. So let's look at the first chemical. The first chemical is acetic acid. Second is butyric acid. Third one is citric acid. And the fourth one is lactic acid. Okay, now here we have the list of only 4 chemicals which are produced in the industries, but already you know many chemicals which you are using in your chemistry labs. Okay, now here look at the list of the chemicals. The first one is acetic acid. Acetic acid is produced from Acinetobacter aceti, Acinetobacter This is name of the bacteria. Butyric acid is produced from Clostridium butylicum. Clostridium butylicum. Again, this is a bacteria. Citric acid is produced from Aspergillus niger. Aspergillus niger. Then lactic acid is produced from Lactobacillus. It is produced from Lactobacillus. Okay, or we call it as a lactic acid bacteria. Now look at the list. Acetic acid is produced from Acetobacter aceti. Now this Acetobacter aceti, this is a bacteria. The name is important because the name of the microorganisms will be asked from which the chemicals are produced. Okay, like the question. Name the microbe that produces acetic acid. Name the microbe that produces butyric acid. So this is how the questions will be asked. You should be able to write the name of the bacteria because as the chapter heading says, microbes in human welfare. So you have to remember the name of all the microbes which we come across the names of these microbes in this topic. Okay. Butyric acid, Clostridium botulinum. Again, it is a bacteria. This is a bacteria. Aspergillus niger. This is a fungus. This is a fungus. Aspergillus niger is a fungus. Lactobacillus. This is a bacteria. This is bacteria. Now, just check out how many of these are bacteria, how many of these are fungus. So, here we have only one fungus and 3 are bacteria. So, this is how the chemicals are produced in industries. So, that's why we call, in short, we call these chemicals as ABCl chemicals. A for acetic acid, B for butyric acid, C is citric acid, L for lactic acid. So that's why remember them in this short form, ABCl chemicals. Okay. So this is how the chemicals are produced from microorganisms. As we come across lot many things that microbes are very essential for the humans because we are using them in large number, in large quantity. What would have happened if these microbes are not there in the nature? We could have not got the chemical. For example, acetic acid. Dilute acetic acid is called as vinegar, which is used in foodstuffs and it produces a sour taste. Okay, as they are widely used in the preparation of sauces, also in the preparation of certain gobi manchurians. So that's why dilute acetic acid is used, which is called as a vinegar. That means vinegar could have not produced if we don't have this Acetobacter aceti. So that's why lot many foodstuffs are also produced from the microbes, as we have studied in the previous classes. So that's why microbes are very essential for the human welfare. Okay, because we are using their products, and these are the microbes, these are the products, and hence we call them as chemicals. Okay, let's go to the next part, enzymes. Now when we come across the enzymes, here we come across different types of enzymes. In the enzyme part, again we are going to study 4 different types of enzymes. So let's study what are those. First one is a pectinase enzyme. This is produced from Aspergillus niger and it is used in clearing fruit juices. Okay, so this is used in clearing the fruit juices. Let's look at the second one. The second one is a protease. Protease. It is used in clearing the fruit juices. Okay, so this is used in clearing the fruit juices. Let's look at the second one. The second one is a protease. Protease. It is used in clearing the fruit juices. Let's look at the second one. The second one is a protease. Protease. It is used in clearing the fruit juices. Okay, so this is used in clearing the fruit juices. Let's look at the second one. The second one is a protease. Now this protease is produced from Bacillus subtilis. Okay, and it helps in tenderness of meat. Okay, then the third one is lipase. This lipase is produced from Candida lipolytica. It is produced from Candida lipolytica. Okay, then this lipase, it removes oil stains and is used in lozenges. Okay, then the fourth one, last one, streptokinase. The streptokinase enzyme. So this is also an enzyme. So all the four what we are discussing are the enzymes. So here it is produced from Streptococcus species. Now this streptokinase is used, it is most important, it is used as a clot buster in myocardial infarction. We call it as MI persons. Okay, now what are these? Let us look at the list of all the enzymes. The first enzyme is pectinase, protease, lipase, streptokinase. So there are four different types of enzymes. Okay, as far as your textual part is concerned, pectinase and protease, they are used in clarification of fruit juices. In your text, they have mentioned in this way. So let's go to the part of the text and then we will come for the additional part. So pectinase and protease are the enzymes. These are the enzymes that are used in clarification of fruit juices in bottle juice. What it means? We also produce fruit juice in our homes, and even the industries also produce fruit juices. The major difference is the fruit juice which is produced in our homes is usually turbid. It is not a clear solution. Okay, but the fruit juice which is packed in the bottles produced by the industries, it is a very clear solution. Okay, as we prepare apple juice or mango juice or even the orange juice in our home, and look at the juices which are produced by the industries. Okay, not by using any of the artificial flavors. It is by the real fruits only. Even though they are produced by the real fruits, this bottle consists of the clear solution. But in our home, we don't get such type of clear solution because industries will use these enzymes that clarifies the turbidity of the fruit juices. Okay, so that's why pectinase and protease are used as a clarification to make a clear solution of the fruit juice. Okay, now this is regarding its general part, but when we come across its particular part, pectinase enzyme is produced from Aspergillus niger. It is a fungus. The enzyme is produced, the enzyme is taken. This enzyme is used in clearing fruit juices, as we have discussed here. The same thing, clearing the fruit juices. Then protease is produced from Bacillus subtilis. This is the name of the bacteria from which the protease enzyme is produced. Now from this bacteria, this enzyme is extracted, and this enzyme is also used in tenderness of meat. During the transportation of the meat gets spoiled or it undergoes the process of hardening. To avoid this hardening, if the enzyme is added, which is if the protease enzyme is added, it leads to tenderness of the meat, softness of the meat. So that's why the enzyme is also used. Okay, next, lipase. Lipase is an enzyme which is produced from a fungus, and the name of the fungus is Candida lipolytica. Candida lipolytica produces lipase. Now this lipase is widely used in laundries to remove the oil stains. This is the most important statement. What is the function of a lipase enzyme? It removes the oil stains. Okay, oil stains. Suppose if there is any oil stain on the clothes, it has to be removed. So how do you remove it? Through the detergents. Okay, that's why lipase enzyme is used in detergents. Which detergents? Okay, like Surf Excel, Ariel. Okay, so all these in all the detergents it is used. So that's why lipase is the enzyme which is widely used in detergents also, and it is especially used by the laundromans to remove the oil stains. Okay, and almost even the lipase enzyme is also present in washing powder Nirma. Okay, so this is getting about the enzyme lipase, which removes the oil stains from the clothes. Then fourth one, streptokinase, which is most important enzyme, which is the life-saving enzyme. Streptokinase is produced from a bacteria called as Streptococcus. Now look at this. It is used as a clot buster in MI patients. MI refers to myocardial infarction, where we also call it as a blockage of the arteries, also termed as a heart attack. MI. Okay. Now what happens here? Suppose if this is the blood vessel, in the blood vessel, if there is any clotting, suppose if there is a clot here, if clotting happens, this streptokinase is used in the form of injections. That is injected to those persons who are suffering with the MI, myocardial infarction, severe blockage. Suppose when the injection is given to the patients, MI patients, then what will happen? This enzyme will enter into the arteries, and when the blood will be flowing here, it removes the clot, it bursts the clot, it removes the clot. That's why streptokinase enzyme is used as a clot buster. Look at the name. It is used as a clot buster because the clot is busted out. The passage is made here. Otherwise, what will happen? If this enzyme is not injected to the persons who are suffering with a severe heart attack, then ultimately it leads to death. That's why it saves the life, and hence given for the MI patients, myocardial infarction or heart attack patients. Okay, so this is regarding all the 4 different types of enzymes. So these are the 4 different types of the chemicals. These are the 4 different types of enzymes. Now let's move on to the last part of the heading, where we come across bioactive molecules. And in these bioactive molecules, we are having 2 names. There are 2 bioactive molecules. Now let's study those 2 bioactive molecules. Now we call them as bioactive molecules because they are active in the biological system. They are more active in the biological system. Now let's write their names. Bioactive molecules. First one is Cyclosporine A. It is produced from Trichoderma polysporin. This is the name of the fungus. Cyclosporine A. Now this cyclosporine A is used as immunosuppressive agent, immunosuppressive agent during organ transplantation, during organ transplantation. Then the second one is statins. Statin is produced from Monascus purpurus. Monascus purpurus. Again, it is a fungus. It is used as blood cholesterol lowering agent. Blood cholesterol lowering agent. So these two bioactive molecules are most important. As the name says, bioactive molecules, they are more active in our biological system, and hence the term bioactive molecules. Then what they do? Cyclosporine A, statin, they are available in the form of injections. As we have the streptokinase here, it is in the form of injection. Similarly, these two are given in the form of injections. To which persons or to which patients? Cyclosporine A is a bioactive molecule which is produced from a fungus called as a Trichoderma molluscum. It is used as an immunosuppressive agent during organ transplantation, like kidney transplantation. What is done here? The person who is suffering with the kidney failure, either he has to go for a dialysis or he has to go for the kidney replacement. When the kidneys are replaced, our immune system do not allow a small microorganism to enter into our body, and even if it enters also, the immune system will kill it. Similarly, body do not accept the larger kidney, and hence what happens? The injection is given, cyclosporine A injection is given to the patient so that the immune system gets suppressed. The immune system gets suppressed so that the body can accept the organ. That's why during kidney transplantation or any organ transplantation, like even heart transplantation also, any other organ transplantation, cyclosporine A is used as an immunosuppressive agent, as it suppresses the immune system so that our body can accept these organs easily. Okay, so this is the function of cyclosporine A. It is available in the form of injections. Statin, again it is available in the form of injection only. Statin is produced from a fungus, Monascus purpurus. It is a fungus, Monascus purpurus. Remember the name of the fungus here. And what does it produce? Statin, a bioactive molecule. For what purpose statin is used? It is used as a blood cholesterol lowering agent. Now what will happen if the cholesterol level rises in the blood? It leads to blockage of the arteries, it leads to heart attack also. So that's why this statin is used for those persons whose blood cholesterol level is high, especially in the aged persons. That's why the aged persons should consume cholesterol little bit less, and they have to control their cholesterol level also. Okay, so this is regarding the part of chemicals, enzymes and other bioactive molecules. So let us end in this session. For more clarity of the voice, use the headphones and listen it carefully. Okay, because this is most important, and each and every heading, term and the microorganism is equally important, because we don't know which will be asked for the exam. And as far as your objective part is concerned, this topic is most important because it deals with the microbes. Okay, so the next continued part we'll see it in the next class.